It's my pleasure to introduce Markus Weingarth. Markus Weingarth was trained in biochemistry in Greifswald in Germany, and did his PhD in NMR with Jeffrey Bodenhausen at the École Normale Supérieure in Paris and the EPFL in Lausanne. In 2011, he joined the solid-state NMR group in Utrecht, the Netherlands, initially as postdoc with Mark Balbus. In 2015, he became independent group leader at the Bijvoet Center for Biomolecular Research and was appointed as assistant professor by Utrecht University. Since 2019, he is associate professor. Markus contributed enormously to building the new high-field NMR infrastructure in Utrecht, making the 1.2 gigahertz NMR and the 800 megahertz DMP system operational. And contributed with visible new solid-state NMR developments, including direct proton detection. In short time, he built an independent research line on studying novel and unique antibiotics and bacterial membranes, aimed at understanding their mechanism. For his research, he received already several awards, including a VD of the Netherlands Science Organization, an ERC Consolidator grant, and very recently in August in Boston, the prestigious Founders Medal of the International Conference of Magnetic Resonance in Biological Systems. He does not forget his students, who also received visible thesis awards for their nice PhD research. Today, Markus will present his latest research, recently published in Nature, with the title Teixobactin kills bacteria with a two-pronged attack on the cell envelope. Markus, the screen is yours.
Thank you very much for the flattering introduction, Rolf. Thank you. Let me begin with thanking Instruct-ERIC very much for the opportunity to present our research on Toxobactin. And since this presentation is certainly a bit on the longer side, I will not only speak on Toxobactin, but I will also show some unpublished data on a brand new antibiotic in the second half of the talk. So let me begin with fixing my slides and a short introduction. So why do we work on antibiotics? I guess this is obvious to all of you. We work on antibiotics because of the antimicrobial resistance crisis. To date, bacteria that are resistant to virtually all clinically used antibiotics are observed routinely, and antimicrobial resistance has become a major mortality factor worldwide. According to a recent study commissioned by the British government, antimicrobial resistance will soon cause more annual deaths than cancer, and already now the annual mortality caused by antimicrobial resistance seems to be on par with the combined effect of malaria plus HIV. So this is serious, and if we want to work against resistance, then we need new antibiotics that use new mechanisms. And good templates for these next generation antibiotics could be so-called lipid targeting antibiotics. And these are antibiotics that specifically target lipids that only exist in bacterial membranes, but not in human membranes. And these drugs are powerful and they kill a broad spectrum of drug-resistant bacteria. In addition, since their lipid targets are conserved and much more difficult to modify than proteins, resistance against these compounds is rarely observed. So you see, these drugs could be great templates to develop next generation antibiotics. However, here comes the big problem. Currently, we cannot efficiently use these drugs to develop antibiotics because we don't understand how they work. So why is it so difficult to understand lipid targeting antibiotics? Well, that's because they use complex supramolecular mechanisms that can only be understood in biological membranes. And to understand these supramolecular mechanisms, we need to study them on several lengths and timescales, from micrometre to angstrom, from hours to seconds. And this requires an array of different biophysical methods with solid-state NMR at the core. And today I will focus on antibiotics that target lipid-2. So what is this, the target lipid-2? So bacteria are surrounded by a network of peptidoglycan that is essential for their integrity. Peptidoglycan is built of strings of repetitive sugars here in blue, that are connected by a short pentapeptide bridges in yellow. And peptidoglycan is in constant turnover, and it is maintained, sorry, this is my coffee machine, and it is maintained via its precursor molecule lipid-2. And lipid-2 carries one sugar peptide head group that is attached to a pyrophosphate group and a long lipid tail. And lipid-2 is assembled in the cytoplasm and then flipped across the plasma membrane, where the head group is chopped off and integrated in the peptidoglycan, and the rest is cycled back for the next round. And this lipid-2 cycle is essential. Any drugs that interferes with it is in antibiotics. This tenet was famously validated by vancomycin. Vancomycin targets lipid-2 at the pentapeptide, and it used to be a drug of last resort. However, you know that peptides can be mutated, and that's why many bacterial strains are resistant to vancomycin these days. Therefore, we work with drugs that have a smarter binding mode. Our drugs target the pyrophosphate. Since this is an immutable target, these antibiotics are ultra robust to the development of antimicrobial resistance, and they're also powerful. A number of exciting lipid-2 binding drugs have been discovered over the last decade, but for none of them, we really understand how they work. And the most famous of these antibiotics is likely Toxobactin, and I'm really looking forward to sharing 2 stories on Toxobactin with you today. But before I come to this, I want to make a point. If you want to study these membrane-active drugs, then you need membranes. So this here is niacin, and niacin is one of the poster childs of the lipid-2 binding drugs. And niacin features 5 so-called thioether rings that are dubbed A, B, C, D, E, and it's well known that rings A and B target lipid-2, and you have here already a beautiful structure that shows niacin bound to lipid-2, and this was solved with solution NMR in mice cells. We have here the original solution NMR spectrum that shows niacin bound to lipid-2 in mice cells. So let's see what happens if we measure exactly the same spectrum, exactly the same complex in biological membranes. And you see, this is a difference like day and night. The spectra are completely different. And this must mean that the complex is also completely different in biological membranes. So you need membranes in order to understand membrane active drugs. What a surprise. And this is something that we published a few years ago. And the first author of the study is Moderator, who's currently doing a postdoc in the home group at MIT. We also have a structure of the complex that is not published yet, but I'm not going to dwell on this today. Instead, I'm really looking forward to sharing our work on Toxobactin with you. So what is Toxobactin? Toxobactin was discovered in unculturable bacteria a few years ago, and its discovery made or hit headlines worldwide. It was globally heralded as a wonder drug, as a game changer in the combat against drug-resistant bacteria. And it is considered as the first new antibiotic discovered in 3 decades. And it has a real perspective. Clinical trials have started. But what is Toxobactin exactly? It is an 11 residue peptide that features many non-canonical elements, including several D amino acids and a so-called Depsy cycle. It's a C-terminus. In the Depsy cycle, we have a very rare funky amino acid called endurosodidine that was only encountered 3 times in nature. And in this original article, it was already shown that Toxobactin targets lipid-2. So what else was known before we started? It was known that the N-terminus is essential for activity. If you chop it off, you lose all the activity. It was also suggested that the N-terminus forms a beta strand in the bound state. And furthermore, it was shown that you cannot change the order of D and L amino acids or the order of polar and apolar amino acids. All this causes a complete loss in activity. So a lot was known before we started, but there were no data in membranes available, and that's what we wanted to contribute. And this was very challenging initially to contribute, because initially we could not work with natural Toxobactin. And that's, remember, natural Toxobactin comes from so-called uncultured bacteria. You cannot simply make this at home. So that's why we initially teamed up with Ishwar Singh, who's here in the audience, a chemist from Liverpool University. And Ishwar is an expert in synthetic Toxobactins, and he showed that this funky residue, endurosodidine, can be replaced by branched hydrophobic amino acids that keeps a good activity, and it slashes the cost of synthesis dramatically. So for the first part of our study, we used these Toxobactin analogs, and to make these analogs amenable to solid state NMR, we introduced 6 carbon and nitrogen labeled L-amino acids that covered the entire molecule. And so here we have already the lipid 2-bound state of Toxobactin in membranes, and we count 1, 2, 3, 4, 5, 6 signals corresponding to these 6 labeled residues. And this is really the bound state. It looks totally different in the free state. And we can confirm that this is a complex using phosphorus solid state NMR in membranes. We have only 3 signals. The big one comes from the bulk lipids and the 2 smaller ones from the pyrophosphate of lipid 2. And once we add Toxobactin, then we see a major shift in the signal of the pyrophosphate, confirming complex formation. So let's look into this complex. And first we wondered about the secondary structure of the complex, or sorry, of Toxobactin in the bound state. Therefore, we acquired this beautifully resolved perispectrum. And from these carbon-carbon spectra, we can readily derive secondary structure by comparing the experimental chemical shift to the random coil chemical shifts. If this difference is positive, it means alpha helix. If it's negative, it means beta strand. And you readily see that the N-terminus adopts beta strand conformation in the bound state. Next, we wondered about the supramolecular arrangement of Toxobactin in the complex. Therefore, we again acquired 2D carbon-carbon spectra with a short and a long magnetization transfer time. With a short transfer time, you see only in intra-residual contacts, and with a long transfer time, you also see inter-residual contacts between several residues. And we're going to compare now the signals in these green boxes that we're going to superimpose. And please focus on signals with red labels. These are the interesting long-range contacts over many residues. And we see a long-range contacts between residues 2 and 6, 3 and 7, and even 2 and 7. And if you consider that the C alpha to C alpha plus 2 distance in a beta strand is 6 angstrom, it would mean that the distance between residues 2 and 7 is 15 angstrom. And NMR people will tell you, well, that's not possible because the maximum distance threshold is about 7 to 8 angstrom. So you can only explain these contacts as intermolecular between anti-parallel beta strands. And we could confirm this oligomerization of Toxobactin upon lipid 2 binding using fluorescent spectrometry and pyrene type lipid 2. Once 2 pyrene molecules come close to each other, they emit a unique X-mer signal at 490 nanometer. And this is exactly what we observed once we added Toxobactin to pyrene type lipid 2. You see the buildup of this X-mer signal in a dose-dependent manner. So together, these data unambiguously show that Toxobactin oligomerizes upon lipid 2 binding. So let's look into these oligomers at high resolution. To determine the structure, we first wondered if the amino proteins of the Dempsey cycle really directly target the pyrophosphate of lipid 2. This was already suggested in the literature, but there was no proof. And to get this proof, we acquired this beautifully resolved 2D proton phosphorus spectrum. And again, the spectrum is quite simple. The phosphorus dimension has only 3 signals from the bulk lipids and from the pyrophosphate. And you see that only the pyrophosphate talks to the amino protons. And to annotate these signals, we need to realize that this proton dimension and that proton dimension are exactly the same. Then we can transfer the assignments, and we see that these signals perfectly match with residues 9 and 10 of the Dempsey cycle. So the Dempsey cycle really directly coordinates a pyrophosphate. And I like to emphasize that these data were not acquired in Utrecht, but in Florence at CERN with kind help of Moreno, you see here. And this was financed by, or supported by Instruct-ERIC. Not only by the conventional access, but also by an Instruct-ERIC awards that Moreno and I shared. So you see us here together at the Chianti meeting that Moreno organized 2 months ago. So this was a great means, not only to get critical data for our project, but also to foster pleasant personal contact. So thank you very much, Instruct-ERIC, for making this possible. So what we also critically need to understand to pinpoint the interface is the roles of the sugars and the pentapeptides. Especially the pentapeptide is important because it's here where bacteria introduce modifications that are known to cause antimicrobial resistance. Just look at the example of vancomycin. In order to understand the role of the sugars and pentapeptide, we needed to carbon-13 label it, and this worked very well. This was again mastered by my former PhD student, Moderator Silver. You see here the sugars of lipid 2, and that's a pentapeptide, and we can use this to unambiguously determine the interface. These here are high quality 2D carbon-carbon spectra in which both Teixobactin and lipid 2 were carbon-13 labeled, and we can derive unambiguous interfacial distances from this. We have here plotted all the interfacial distances on the primary structure of Teixobactin, and you see that the contacts cluster on the Debye cycle. The Debye cycle is indeed the binding interface, and you also see that almost all contacts relate to the Mernek sugar that is directly attached to the pyrophosphate. So Mernek must be directly at the interface. There were hardly any contacts with the Glücknack sugar, which must be distal from the interface. And then strikingly, we did not observe any contacts for the pentapeptide in the so-called dipolar spectrum. And this suggests that the pentapeptide is highly mobile, because you can only observe rigid residues in this so-called dipolar spectrum. And to confirm the mobility of the pentapeptide, we acquired a so-called scalar spectrum. And a scalar spectrum, conversely, you only observe highly mobile residues. And here, indeed, we only observed the pentapeptide, confirming its mobility in the complex. So let's put this all together. Thus far, we know that the Debye cycle coordinates a pyrophosphate, Mernek is directly at the interface, Glücknack is distal from the interface, and the pentapeptide is highly mobile and not at all involved in the interface. And that the pentapeptide is not involved in the interface is the very reason why Toxobactin is so robust to resistance development, because it doesn't care for all the modifications that bacteria introduce in the pentapeptide. Now, the last thing that we needed to understand in order to determine the structure of the complex was the membrane topology. And here we used an old but gold trick from the group of Mei Hong. It's a so-called mobility edited experiment where we transfer magnetization from mobile water and lipids to the rigid antibiotic. And you see that the rigid antibiotic talks to both the water and the lipids. This is characteristic for a compound that sits at the water membrane interface. The exceptions were these long hydrophobic isoleucines that only talk with the lipids, and thereby we know the membrane topology. This is the structure of the complex. You see here in yellow the antiparallel Toxobactin beta sheet, and you also see that the amino proteins of the Debye cycle tightly coordinate the pyrophosphate. But what I like most about the structure is that it explains the complex stereochemistry of Toxobactin. In the complex, we have a clear-cut separation of hydrophilic and lipophilic residues. Lipophilic residues face the membrane, hydrophilic residues face the water. And this distribution is only possible with this stereochemistry. If you imagine it a hypothetical all-L Toxobactin, you would have some hydrophilic residues that face the membrane and some hydrophobic residues that face the water. So a favorable distribution is only possible with this stereochemistry. So that's what we published 2 years ago together with Ishwa, and we made one more quite interesting discovery. So we knew that lipid 2 oligomerized upon, or the Toxobactin oligomerized upon lipid 2 binding. And we wondered, hey, can we even observe these oligomers on the macroscopic level with fluorescence microscopy? Therefore, we used MBD tagged lipid 2. And you see here the uniform distribution of this tagged lipid 2 on the surface of the liposome. And once we added Toxobactin, then we observed these striking, massive micron-sized clusters. And that was highly surprising, but we had no idea what this meant. So at the end, there were two open questions. First, what is the role of these clusters? And second, what is the role of endurocytidine, this funky residue that was only observed 3 times in nature? And then we were lucky. So on the day that we published our article, I got a phone call from Kim Lewis, and Kim, together with his company Novabiotic, is the discoverer of natural Toxobactin. And after some talking, we soon had or received a few milligram of uniformly carbon and nitrogen labeled natural Toxobactin. And as you see yourself, natural Toxobactin bound to lipid 2 in membranes gives fantastic data that we can readily completely assign. We have here the funky residue and endurocytidine, and this is D-N-methylphenylalanine at the N-terminus, which is also important, as I will show you later on. So long story short, also natural Toxobactin forms antiparallel beta sheets, and we can also readily determine the interface using carbon-13 labeled lipid-2. If we zoom into the enduracytidine side chain in red with unlabeled lipid-2 and in blue with carbon-13 labeled lipid-2, and all these blue signals without red correspondence provide us with unambiguous contacts between enduracytidine and the Myrmic sugar of lipid-2. So here we have plotted all the interfacial contacts in the primary structure of natural Toxobactin, and you see that the interface is completely dominated by this funky residue enduracytidine, and this has consequences for the binding energetics. So if we compare this with the leucine 10 analog, we see that natural Toxobactin has a higher affinity and also a more favorable binding enthalpy, and a more favorable binding enthalpy means a higher specificity due to more favorable hydrogen bonds and more favorable electrostatics. So if we pull this all together, then we see that natural Toxobactin tightly, rigidly binds to the sugars of lipid-2 because of the presence of enduracytidine, whereas the leucine 10 analog has a more fuzzy binding interface. So this means that that's the reason why natural Toxobactin has a higher affinity and a higher specificity. So this here is the structure of the complex. In blue, we have the antiparallel Toxobactin beta sheet. We see that the Dempsey cycle tightly, specifically targets a pyrophosphate, and enduracytidine forms out hydrogen bonds with Myrmic. And then strikingly, we also observed that the cationic end terminus of an adjacent Toxobactin directly coordinates a pyrophosphate. So every lipid-2 is doubly coordinated by two Toxobactin molecules in the complex. And this is one of the reasons why Toxobactin needs to oligomerize for efficient lipid-2 binding. So thereby, these data explain the role of the non-canonical elements in natural Toxobactin, and this answers one of the two open questions. But what about the other major open question? What about these clusters? So natural Toxobactin also forms these clusters upon lipid-2 binding. You see this here with fluorescence spectroscopy. And strikingly, this time, we observed these membrane perturbations at the side of the clusters. You see this even better in these bright field images. And these membrane perturbations are in agreement with static phosphorus solid state in the Moderator that are sensitive reporters on the state of the membrane. You see that after the addition of Toxobactin to liposomes, the state of the liposome changes. That means there is some perturbation in the lamellarity, regularity of the membrane. So this is interesting, but this is all in liposomes. So does this really happen in bacteria? Does this really contribute to Toxobactin's killing mechanism? In order to address this, we teamed up with James Norvig from UC Urban and Harold McGillivray from Utrecht University. James provided us with the fluorophore Toxobactin. And you see that after the addition of fluorophore Toxobactin, we also observe these massive clusters that unambiguously shows that cluster formation is a part of Toxobactin's killing mechanism. And this happened really rapidly, 15 minutes after the addition of the stacked Toxobactin. So thus far, we have established the nature of these clusters at the submicron level and at high resolution. But to truly understand the nature of these clusters, we needed to close this resolution gap. And we did this with atomic force microscopy. And we did this in collaboration with Walter Roos from Groningen University in the Netherlands. And these data are pretty clear-cut, so I will rush through this. You see that Toxobactin and lipid-2 form these fibrils. This is in perfect agreement with the solid state and the mass structure. And you see this very nicely in this video. So once we add Toxobactin to lipid-2 doped membranes, within about 10 minutes, we see the emergence of these fibrils. And what is also interesting is that this timeline, so 10 minutes after the addition of Toxobactin, matches very well with the emergence of clusters in bacteria that we observe after about 15 minutes. So Toxobactin forms fibrils together with lipid-2. And the last points that we needed to address were these membrane perturbations that we observed at the site of the clusters. And here we need to realize that AFM is a gorgeous method to measure the heights of the membrane, that you can do it almost at atomistic resolution. So we're going to look now at the heights of the membrane at the site of these clusters. This is here in dark orange, and here in dark blue. So you see now the heights of the membrane, and you see that at the side of the clusters, the heights of the membrane is strongly compromised, thinned by almost 1 nanometer. So what happens here? What causes this membrane thinning? And we try to rationalize this with atomistic MD simulations that were done by Francesca from my group. And also here we could reproduce this membrane thinning very well in nice agreement with the AFM data. So what happens here? On the one hand, the strongly hydrophobic underbelly of Toxobactin pushes polar lipid head groups away. And on the other hand, and presumably more importantly, in clusters, we naturally have a very high concentration of lipid 2. And lipid 2 has this strongly non-lamellar, super long C55 tail that you also see here in these simulations. And this tail reaches out into the other layer of the bilayer, and this compromises the membrane integrity. And indeed, in membrane permeabilization assays, shortly after the addition of Teixobactin, we see that the membrane potential of bacteria disappears because Teixobactin compromises the membrane integrity. So let's put this all together. At the membrane surface, Teixobactin settles and targets lipid 2, oligomerization into these fibrils. And first, these fibrils blocks the peptidoglycan synthesis. And second, these fibrils compromise the membrane integrity. And the great thing is that these fibrils only form in the presence of lipid 2, and lipid 2 only exists in bacterial membranes, and thereby Teixobactin elegantly solves the selectivity and toxicity issue. So this is something that we published recently, and the first author of the study is Shukla, who did really a wonderful job and contributed to almost all methods in this multidisciplinary study. And she stands here in front of our 1.2 gigahertz magnet. I will show you some data from this magnet later on. This here is a nice and admittingly a little bit over the top illustration. So I don't think that these fibrils really rupture the membrane. You can rather imagine this as a perturbation instabilization of the membranes. And this illustration was done by Jan Bart van Rossum from the FMP Berlin. Thank you very much for this again, Jan Bart. With this, I'd like to come to the acknowledgements. So I have already thanked many of our wonderful and inspirational collaborators. I want to single out Kim Lewis again, because without his support, we simply couldn't do much or many of the studies that we have shown. And then I'd like to thank my group again. So he has written the first author of the Clovibactin and Teixobactin stories. This is Mike, the first author of the plexes and manuscripts that we hopefully soon submit. And I really would like to thank these wonderful young and inspirational people again. So it's really great to see how their expertise and passion drives these projects. I also like to thank my collaborator, Evian again, who is essential for the lipid 2 works that we do for plexes and Teixobactin. Tanya again for her wonderful contributions to the Clovibactin paper, and Ishba, who was instrumental for the first part of the story, Wouter for the essential and excellent AFM studies, and then eventually Instruct again for the invitation and you for your attention. Thank you for listening. Thank you.